My name is Sijbrand de Jong. I'm the Dean of the Faculty of Science of Radboud University, and on my tour through the faculty I'm now at the Radboud Institute for Molecular Life Sciences. So we are here with Jorine and Rosemary, both from what we call RIMLS, and working on everything from molecule to man, and they're working on different aspects. Jorine, you're working on the more physical aspects of life. Can you explain us what you're doing?
I am fascinated by the process of how DNA fits inside human cells. So there's 2 meters of DNA in every human cell, and this somehow needs to be folded into a cell, a container that's orders of magnitude smaller. And there's a lot of physical processes behind that. So I'm interested in figuring out how those physical processes lead to the organization of DNA and structure and function.
And for this you use a piece of equipment which is brand new. Yeah. There's hardly any speckle of dust on it yet. And it's a microscope. And of course, for most people, a microscope is 2 looking glasses. But this one looks quite different.
Yeah, it indeed is very different. This is a tool where you can look at single molecules. So it is an optical trapping microscope. So with this, I can look at single molecules of DNA and sort of trap them with like little tweezers and pull on them and look at the structure of that DNA. I expect lots of fun science, starting with this new microscope, which is the only one in Nijmegen and in the Netherlands and in Western Europe. So I'm excited to do some really cool experiments with this piece of equipment.
Very interesting to just have the inside of the DNA. But then, of course, this DNA is used to express itself in cells. And now we go to Rosemary. Hello. Because you're working on?
So my lab takes a systems biology approach to study the influence of gene expression and metabolism on cell behavior. So gene expression and metabolism, both of these are very complex networks. Each of them involves tens of thousands of components, right? So this is transcripts and proteins and metabolites and so on. And the difficulty in studying them has been, if you manipulate a small part of the gene expression or of metabolism, the entire network will change in response to that. So it's really difficult to study, and to tackle this problem, my lab takes a combination of wet lab and dry lab approaches. So wet lab means experimentation, so we go into the lab and we manipulate our cells in very specific ways and we collect data. And dry lab means computational modeling, and so to do this we are plugged into the Dutch national supercomputer Snellius. And we use computational modeling to predict cell behavior. And this can help us to understand how cells behave in a healthy individual. And also this helps us to understand how diseases related to gene expression and metabolism come about, and also helps us to simulate potential therapeutics to help fix these problems in diseases.